name's Hope, and this is my special guest, Victoria.
And today, we're going to be discussing science in the news.
More specifically, we are going to discuss the accuracy of popular media outlets and their reporting of scientific findings.
To do so, we will investigate a story by the New York Times that highlights a recent study on species of toads in Australia.
The news story talks about the latest study on cane toads, which are native to South and Central America but were introduced to Australia in the early 1930s. The main topic of discourse in this story is evolution, as this species of toads has demonstrated cannibalistic behaviors over the last few decades.
Cane toads are a large, invasive and poisonous species, and they have been studied recently as scientists have found out that tadpoles devour their youngsters' developing relatives.
The news story references this phenomena as evolution in fast motion, as these toads have been observed to be demonstrating this cannibalistic behavior over the last couple decades since arriving to Australia.
The toads only demonstrate this sort of behavior in the Australian environment, which is why researchers are saying why we are witnessing evolution happening in real time.
According to the news story, a scientist in Australia named Michael Crosland conducted a study 10 years ago to investigate the impact of native frogs when he found that cane toad tadpoles have a fierce appetite for other hatching cane toad eggs.
This led to the current study discussed in the news story in the primary literature, recently published findings that found why the cane tadpoles feast on eggs and hatchlings.
It was revealed that the toxin known as bufadienolide was the culprit for the fierce cannibalistic appetite found in Australian cane toads. This toxin is the same chemical that makes the frogs poisonous.
In the story, it is stated that the tadpoles that were exposed to the bufadienolide toxins ate native frogs' eggs and frogs' eggs of their own with great pleasure.
The New York Times article generally explained the bufadienolide was the main cause for the behaviors demonstrated in this toad species and kept the details on the experiment simple.
The primary literature article itself reveals more about the trials that were run and gave additional background information about cane toads.
The researchers conducting the study ran a total of 3 main trials, and the materials and methods used are explained in great detail with figures in the primary literature article. The first experiment tested toad tadpoles as predators of frog embryos versus toad embryos.
Results were insignificant as cane toad tadpoles ate fewer eggs and hatchlings of the native frog species compared to others within the same species.
The results were deemed insignificant because the presence of the tadpoles did not majorly reduce the survival of the native species.
In experiment 2, the toxins was introduced to the test the effect it had on stimulating cannibalisms of toad eggs and hatchlings.
Results were significant because a large portion of the toad eggs and hatchlings were cannibalized in the presence of the toxin treatment. In fact, it was found that the chances of the eggs and hatchlings being consumed in the presence of the stimuli was 3 times greater than those not exposed to the stimuli.
It was also determined that time played a factor in cannibalism, as eggs of conspecifics were more likely to be consumed over time if the toxin was present.
In the final experiment, the effect of bufadienolide cues on predation on frog eggs was tested.
This was done using the tadpoles and L. tornieri eggs and 2 controlled treatments that were used and an inert ceramic ring and a toxic ceramic ring.
It was found that predation rates of the unhatched eggs were low in the absence of the toxin cue and high in the presence of toxin cue, no matter the prey species.
After reviewing the study itself, a few areas of inaccuracy within the news story were found. For example, it was stated multiple times that the cannibalistic toads that had great appetite for their young developing relative.
In the news story, it was stated that when exposed to the toxins, the tadpoles consumed native frog eggs and those of their own kind with gusto. This is somewhat inaccurate because in the primary literature article, the trials conducted failed to have significant results that proved the fierce appetite that is exaggerated in the news story. Yes, the tadpoles did end up feasting on younger developing relatives when exposed to the toxin, but it was nowhere near as significant or fierce as described by the New York Times.
The news story was also inaccurate because when describing the cannibalistic tadpoles, they kept mentioning how they ate the eggs and hatchlings when exposed to the toxins.
We found this to be another area of inaccuracy because in the second trial conducted, it was found that there was more of an appetite for the eggs specifically over time, which led the focus of the third experiment to be with a specific species of egg embryos rather than eggs and hatchlings.
One point that is accurate though is that the behaviors that are being demonstrated are 100% allowing us to see the evolution happen in real time.
These cannibalistic behaviors that have been occurring over the span of decades are showing researchers that cane toads in Australia are evolving to consume those of their own species to reduce the number of cane toads in their habitat.
More studies will be conducted in the future and over time to make further observations in regards to how the cane toads are evolving due to this toxin and cannibalistic behaviors.
Future studies will also observe how hatchlings are evolving to defend themselves over their older relatives as the behaviors demonstrated have evolved over such a short period of time.
In conclusion, the accuracy of popular news media outlets can have areas of inaccuracy while still proving to be inaccurate in other areas. It's best to read the primary literature to get most accurate and non-exaggerated amount of information possible.
Thanks for watching!